The overall goal of the TTFields in vitro application system is to investigate their effect on cancer cells from different tumor types, to guide TTFields clinical administration and optimize treatment outcome.
This method can help define the effect of different TTField parameters on cancer cells. The main advantage of this technique is that it allows for preclinical identification of the most effective TTField frequency that inhibits growth of a specific cancer cell line, which could be ultimately applied in humans. By slight modification of this technique, it is possible to test the efficacy of the combined treatment of TTField and other treatment modalities, such as chemotherapy agents and radiation therapy. Demonstrating the procedure will be Yaara Porat, a researcher from our team.
To begin, install TTFields dishes with covers onto a base plate, and prepare an additional 8 dishes that will be used for growing control cells. Place a sterile 2 mm cover slip on the bottom of each dish. Next, suspend 5 × 10⁴ U87 Mg cells in 1 ml of DMEM medium.
The appropriate number of cells plated in each dish depends on the properties of the cell line in use. It is crucial to calibrate it prior to the experiment in order to avoid cell overgrowth.
Pipette 200 microliters of the cell suspension onto each cover slip, such that a drop is formed on its surface, and cover the dishes with their lids. Incubate all dishes at 37 degrees Celsius 5% carbon dioxide overnight to allow the cells to adhere.